And then we use a whole genome amplification to get enough amount of DNA. And then we use a standard exome library preparation kit from Agilent. And then we perform the HiSeq 2000 sequencing. And at last, we did the bioinformatics analysis. This is a whole pipeline of our single-cell genomic analysis. There are two major questions often asked by our audience. First is how we isolate each single cell. In the studies I will introduce, we just use a microscope, and we pick individual single cells by hand. This is the easiest and cheapest way. But here, we choose isolate single cells under the microscope by hand because we want to use this concept because we random isolate the single cells. We try to use our technology to distinguish the different cells, no matter the cancer cells or the normal cells. And other methods like FACS is also okay. But here we recommend you better use a cell surface marker because we all know that some markers in the nuclei will influence the whole genome amplification processes. If you have experience of this, you may know that. Another question is how to amplify the whole genome of a single cell. Here we choose a method based on the Phi29, which is, I think, quite familiar enzyme. And then we added the random primer and perform the whole genome amplification. So what is the performance? Here is a result of our method and the PCR-based method. From the left picture, you can see that we can get a larger amplicon fragment size than the PCR-based method. And this is, I think, very important factors because if you use a PCR-based method, you can only, no matter how deep you sequence, you can only recover part of the genome because of the limitation of the principle of this method. But if you can obtain larger amplicons, it is easier for you to recover most of the genome of a single cell. And then we evaluate our method by sequence two single cells. One is both of them from the first Asian genome donor. We call it YH, and we also sequence millions of cells as control. So we first have to evaluate our methodology. And here we can see that our single cell method can recover more than 95% of the genome, which is comparable with the control data. And then from this picture, you can see if you sequence more than 10x of the genome, you can recover more than, I think, more than 20% of the genome, which is comparable with the control. But here, under the 10x, you can see a bias, and this is accessible because of the whole genome amplification. And then we try to give a whole picture of our single-cell sequence methods. From this circle plot, you can see the blue one, the two cycles of the blue one. Sorry? Oh, sorry. Yeah, the two cycles of blue one is the genome coverage of our single cell data. And you can see our data can recover most regions of our genome. But here we can see a significant GC content impact of the whole genome amplification processes. So this gives us a suggestion that if we want to optimize our method, we can think about the GC content of the genome region. And after the whole picture, we have to evaluate some major artifacts during the whole genome amplification. First is limited template amount will lead to the allele dropout. That means if you have a heterozygous loci at a gene, but since we have only 2 copies of DNA, it is very easy to lose one of them. That is allele dropout. And the other one is the enzyme may make mistake during the long and hard work, as I displayed. Because we can get more than 20 kilobases amplicon. So that is a false positive mutation here, which I showed in the right picture. And then we have to calculate what is allele dropout rate and the false positive mutation rate. Here, the allele dropout rate is about 20%, and the false positive mutation rate is very low, which is comparable with the previous report. Here, I think you may understand that this allele dropout is quite high, as many other scientists asked us. But if you have more than 5 or more than 10 single cells, if you look at a specific loci, and you can easily reduce the influence of the allele dropout. And then we have to evaluate whether the amplification artifacts will show preference on different base types, because this is very important for cancer study. Here we can see that the allele dropout show no preference here, and the amplification error have a strong bias at C to T, G to A, and this is acceptable because it is much easier for C changed to T and G changed to A, as I think if you have a normal tissue, you cause a germline mutation. This is also the most common type. Last, we have to evaluate whether our amplification processes will affect the biological processes analysis. Through our analysis, we can see that no bias could be derived here. So then we apply this methodology to the tumor study. Before we start the project, we have asked some questions like the samples limitation and heterogeneity and cancer progression. And these 3 factors is the most important area of cancer research now. Here is a story about the Tibetan. We presented, I think, 2 years before on Science. And this is a very interesting story because here, this is 50 Tibetan individuals and 40 Han Chinese individuals. If you do not did the genetic analysis, you could not know what is the difference and what is the difference of the gifted ability to the high altitude. And then we use exome sequencing to sequence these 90 patients. By the population genetic analysis, as my colleague QB just told you, he told you much about the methodology of this method. And we first can build the phylogenetic tree, and we can easily distinguish the Chinese people, the red here, and the Europe people, and the African people. After that, we can use SMP information. If you notice this picture, this is quite the same as QB just told you. And use this picture, we can see the Tibetan and Han Chinese are genetically very similar, but there are some significant difference sites, like this gene. Besides this, we can also infer the population evolution history of these 2 kinds of populations. And then we can also build a mathematical model to distinguish these populations further. Here is an example of the population branch statistic, which is we collaborated with Rasmus Nielsen at UC Berkeley. And here we can see, after the mathematical calculation, we can see these three genes show significant difference between these two populations. And here B, if you use all the SNPs to distinguish them, you can hardly make it. But if you use those significant loci, you can easily distinguish the Tibetan people and the Han Chinese people. So after this analysis, we can find that those 50 Tibetan individuals and 40 Han Chinese individuals, and we identify the gene showing strong selection signal here. And of course, we did the further functional validation, and we found this gene is associated with the hemoglobin level and the expression level difference in placenta. So this is a very successful case for the population genetic study. And then, if we try to use this analysis to the single cell tumor study, maybe something interesting. So these are some important aspects like the heterogeneous cell population, cell lineage analysis, development history, and the key genes to tumor. I think which is the same question we asked in the human genetics population analysis. So we started the large single-cell exome sequencing project. First, I will give you an introduction about the first one, cancer progression inferred by 1,000 single-cell exomes from 5 cancers. And here is a 2-cell paper we published early this year. Also, I will add 2 other papers which are in preparation. This is our data set, the kidney cancer, bladder cancer, gastric cancer, colon cancer, and 2 kinds of leukemias. I will not show you the details because of the limitation of the time. And here, I choose 4 cases which I will give further details. And please remind this ET is a JAK2 negative patient, and this kidney tumor patient is a VHL wild type, and the bladder tumor is a muscle invasive type, and the colon tumor here have a large patient cohort. First is data generation. We can make a conclusion from this table that we can recover more than 70% of the exomes of each individual single cells for our later analysis. And then we built the analysis pipeline, including these factors, and the main procedures include somatic mutation calling. And here we have to take the amplification artifacts into consideration. And if you try to know more about this, you can read the supplementary information about the cell paper. And then is the population analysis. And last is a progression inference and identify the key genes. Just to give you some examples, like this ET is a JAK2 negative patient. First, we infer the heterogeneity of this tumor. We distinguish the cancer cell and normal cell by the principal component analysis. But interesting, in the kidney tumor, we found that these 3 yellow cells, they are mixed with the normal cell, which here, the 3 yellow cells were cells we randomly picked up from the tumor tissue that the doctors gave us. So this is quite interesting. We first use a genetic analysis to distinguish the normal cells in the tumor tissue.